Welcome everyone. Today we're doing an interview with Dr. Brady Carter, one of the leading scientists when it comes to water activity. And we want to talk with him a bit about the pharmaceutical market. Welcome Brady, and thank you for joining us today.
Thank you.
So, why hasn't water activity not being a leading parameter in the pharmaceutical market?
Yeah, I mean, I think it's because it hasn't been traditionally used within the pharmaceutical industry, unlike the food industry, where it's had widespread use for many, many years. Within the pharmaceutical industry, it hasn't been part of official standards or regulations, and so it just hasn't gained a lot of interest. Even though all through the time that it's been used in food, it could be used in pharmaceuticals as well, because it essentially provides the same levels of safety and quality protection as it does in the food industry.
So we see in the food industry a lot of use cases where water activity is being used as a very relevant parameter to measure for food safety and food quality. As you mentioned, it's now gaining a bit more interest in the pharmaceutical market. So how does it come that now is the time that they start to look more into water activity and that it's being more widely used in this sector?
Yeah, I mean, I think it's the first thing that occurred that helped with being able to better utilize water activity in the pharmaceuticals was the publication of USP 1112 in 2006, and in that was an informational chapter put out by the USP, and in that chapter it did give some guidelines and recommendations for utilizing water activity for several different applications, including for stability of active ingredients, for excipient selection, but probably most importantly, it was for justification for reducing microbial testing. And so that informational chapter came out, and so that really started some interest within the pharmaceutical industry to learn a little bit more about water activity, and for some companies, they implemented water activity testing. But that USP 1112 wasn't an official method, it was just an informational chapter. Then in 2021, though, we had the publication of USP 922, and that's an official method from the USP for water activity. So it talks about how to measure water activity, it gives information about validation of water activity meters, and still provides those justifications for its use in applications for being able to help with safety and quality.
So you just told us in the USP there are also like some of the uses it has in the pharmaceutical sector where it can help with. Can you tell us a bit more about with what it can help with?
Yeah, for sure. So listed within within USP 1112 as well as USP 922 are some recommendations for applications, and one of those is stability of active ingredient. And the way that water activity helps with that stability of the active ingredient is that those active ingredients become susceptible to potentially to degradative chemical reactions. And water activity is related to reaction rate of most chemical reactions, and especially the ones that typically would be responsible for breakdown of active ingredients. And so by controlling water activity to specific ideal levels, you can maximize that stability for the active ingredient. And so that's really a critical piece of what water activity can do, because that active ingredient is the most expensive part of the pharmaceutical product. In addition, it talks about excipient selection, and the purpose of excipients as bulking agents within pharmaceutical products is that they also serve as a protector for the active ingredient. And one of the ways that they do that is by preventing its water activity change or changing of the active ingredient by preferentially taking moisture. And so in most cases they want those to be hygroscopic, and that's a property of how the water activity changes when moisture is taken up. And if they can take up moisture without having big changes in water activity, then it prevents the water activity of the active ingredient to change as well, and so it makes it remain at an ideal water activity and then maintain its stability. Another thing is for handling of excipient powders. So these excipients are typically amorphous powders. They may be crystalline powders as well, but in either case, water activity is associated with their stability and keeping them from caking and clumping. And this comes from what's called a critical water activity, where they will experience a phase or state change. In these cases, it's for amorphous powders, a phase change. Glass transition that can then result in them starting to cake and clump, and that makes them hard to handle, hard to tablet, changes their dissolution properties as well. And so by identifying a critical water activity and keeping it below that, you can maintain the powder and flow. And then the of course, main emphasis for water activity is its ability to be able to stop microbial growth. And so within the USP 922 and 1112, there is justification for reduced microbial testing. And this is because if you make a pharmaceutical product, you process it with good manufacturing practices to water activities less than 0.7, there's no chance for any growth to happen that would then lead to an infection. So as long as you process it properly with good practices, and then you measure the water activity and confirm that it's below 0.7, there's really no need to do additional microbial challenge testing because it just can't support growth. In fact, it's the most effective way to show that there isn't going to be any contamination because it's measuring an intrinsic property of the material that in of itself prevents the growth of microorganisms. So by doing this, you can drastically reduce any kind of microbial testing for products whose water activity is below the growth limit for microorganisms. And that saves tons of money.
Yeah, and I mean, that's what people want to hear, how they can save money, right? So if I can like just collect my thoughts I got from what you were saying is like it helps with the chemical stability so that you have better product quality, especially with caking and clumping, so you don't get these problems and get happier customers in the end, as you don't have to do any recalls and stuff like that. And also you can reduce microbial testing, which means at the end of the day, like you just said so nicely, it will save money. So just to conclude the whole thing, what would your recommendation be for someone or for some company if they want to start testing water activity for their pharmaceutical products?
Yeah, and for sure, and it's the good news is that it's real easy to get up in testing for water activity. The instrumentation is very easy to use. We have the LabMaster Neo right here. It's a top of the line water activity instrument. You basically plug it in, put your sample in, and push start, and that's all there is to it. So there's not a lot of training or anything that's needed to start measuring water activity. You just simply can implement it. So to start measuring water activity, you just simply need to contact a local distributor about the water activity instrument and start testing. And then start implementing it into release specifications and your standard operating procedures. So it's a really powerful tool that's easy to implement.
Perfect. That concludes our interview. Thank you very much, Brady. If you want any more information, let's check out our application note about pharmaceutical products with water activity as well. It was written by Dr. Brady Carter, actually. And yeah, we hope to hear from you, see you soon, or let us know if you have any questions, or also if you have some questions for Brady. We're happy to connect you with him. Thank you very much.